Okay, this is the last section of cell cycle, which is Unit 6. And this depiction actually shows the cell cycle. And if you take a look at between this black line, which says cytokinesis, the end of the cytokinesis all the way through G1, all the way through the S phase, which is DNA synthesis, all the way through G2, this is what's called interphase. And this is when the cell's doing what the cell's supposed to do. It could be a muscle cell, could be connective tissue, skin, hair, follicle, or anything. The very small section here is what we just talked about, the P mat. Here's the I, P mat. Here's the P mat and the C. This is the mitotic phase, which is called the M phase. And then right after mitosis, right after the cell goes through cytokinesis, whether it be a plant cell or an animal cell, it enters G1. This is a growth stage, and this is where the cell actually grows all the extra organelles as well as cytoplasm that it's going to need. Here's where we actually make the second copy of the DNA through DNA synthesis, and then we actually do check the cell and make sure it's grown enough, make sure the DNA synthesis was done okay, and a little bit more growth, and then we go through mitosis again and start the whole section over again. This is the cell cycle. Here's another one which sort of explains it. Here's the PMAT. This is the I interface, and G1 where we actually get cell contents excluding the chromosomes are duplicated, so all the organelles is what is cytoplasm. Here's where we actually make the 23 chromosomes into 46 chromosomes. So we can actually go through mitosis again, and here's where the cell double checks all of its duplicated chromosomes as well as all the repairs that it needs, and if that's true it goes through mitosis again, and that's the cell cycle. So I, prophase, metaphase, anaphase, telophase, and then cytokinesis, and then start the whole process again. There's also a GO where the cell cycle will arrest or stop if it actually finds out that the mitosis did not go correctly. And the only time this actually goes bad is if we enter the cancer area. So cancer does not go through G1. Cancer just keeps going through even though the cell knows that it's not working well. Here's the same section where we actually go to GO. We can maybe fix it and it can go back into G1 where we actually prepare for synthesis, doubling, same sort of thing, the cell cycle. Here's the same thing, except we're actually putting in ATP to ADP, and we're also talking about some of the proteins and enzymes that go into the whole system. So you can see we actually have lots of different things. And then if it goes to the GO, we actually degrade it into its parts if we can't fix it. Real pictures of the cell cycle in animal cells. So here's an animal cell in interphase. There's the diagram. We actually go through early prophase, and then late prophase, and then metaphase, and then anaphase where they separate, and then mitotic telophase where we actually start making the, and this is an animal, the cleavage furlough. And here it is in an animal cell, interphase, prophase, metaphase, telophase to anaphase, and then there's the formation of the cell plate. Onion root mitosis. The onion root, because it's going through soil, has to produce a whole lot of new cells. The cells out here, these are epithelial cells. These actually die, and as it hits the new sediment, these get loughed off. So it's producing these cells at a phenomenal rate, as well as the root itself is actually growing. And these again are the roots of an onion cell, which grow really well. Here's a picture, and if you take a look, we can see lots of cells in interphase. We can see a couple of them actually being relatively small. These are actually just completed mitosis. You can see anaphase, you can see metaphase, you can see there's two more that are actually splitting for anaphase. Maybe cell plates in a couple of these. This one's a little early, but you can see lots of cells minding their own business, doing their own job. Okay, this is a very large picture, and if you go through, you can actually see, especially down here at the bottom, you can see lots of cells actually going through. There's metaphase, there's an anaphase, there's can't really tell what phase. There's another one going through a phase. You can see cells that have just already split, starting to make the plate in the middle. I wish I could zoom in a little bit more on these, but you can go through and actually see most of these cells look normal. There's another anaphase, another metaphase, metaphase, but a really cool picture. Okay, here's a animal, I'm assuming a plant cell, which is actually going through lots of things. You can see most of the cells look kind of normal, but we can actually start making the chromosomes. We can line up the chromosomes in the middle. We can pull the chromosomes apart, and we can actually start making a new cell plate to create two new cells. And extras, pinocytosis, where we actually bring water into the cell. Phagocytosis, where we actually start bringing food into the cell. And homeostasis, where we actually maintain the content or the internal environment of the cell. And that's what the cell membrane is trying to do, is control the flow of things in and out of that cell to maintain the cell in peak form. And then inner life of cell, full version, I'm going to show this one to you. We're in a blood vessel. It's a white blood cell. The underside of the cell membrane, where they latch on and release so they can move. There they are, grabbing onto each other. And now they're just kind of staying on to the same place. Cell membrane, fluid mosaic, rafts that float through. This is a lipid, a fat raft. Lots of protein markers sticking up. Cholesterol, kind of giving the cell rigidity so it's not too fluid. You can see proteins going through the entire cell. Lots of enzymes floating around, grabbing onto things. Cytoskeleton. Protein, channel protein. Here's another protein shown by the squiggly ribbons that are on it. This one's only on one side of the cell. More cytoskeleton, more proteins. Inside the cell membrane, these cytoskeletons give rigidity and form. Lots of moving things around the cell membrane. Cytoskeleton, which can actually act as a highway inside the cell, highway system. Creation of them. And the destruction of them by a protein. And then they unzip. Then we have microtubules forming. That'll be like a centriole. And then they also, when they get no longer utilized, proteins pull them apart, lyse them. Along those tubules we actually get vesicles with proteins, and the proteins walking along carrying these things from point A to point B. Called a motor protein. Mitochondria, the powerhouse of the cell. Cellular respiration, sugar and oxygen going in and ATP coming out. There's the centrosome. Aster kind of view. Notice there are lots of mitochondria, bacterial type cells. This is the nuclear membrane, where we have RNA going out, meeting a ribosome, and then it gets read, large part, small part, and then we get this new protein coming out. And it could be a motor protein, could be a protein for the cell membrane, could be a protein to make anything else. This is a protein going to the cell membrane and actually producing its protein, or a ribosome going through making the protein, so it actually goes from one side of the membrane to the other. Actually, this is going to be an intercellular protein. Here's the endoplasmic reticulum. There's another motor protein, another vesicle. And all this is going on the inside of all your cells. That's a Golgi body. That's kind of cool to watch. Packages material for inside the cell or extracellular transport. They do like those motor proteins. Plasma membrane. It's either endocytosis or exocytosis. Looks like it's endocytosis. Brought material into the cell. Chemokine, actually it's showing you, and that's a receptor. Proteins that they're identifying, I'm not sure what a G protein does. If you're going to get into cell biology, this is the kind of thing you're going to be talking about. Cell skeleton, cytoplasm, and then the cell membrane. Proteins actually doing jobs. There's that white blood cell again. Moves along inside the blood vessel, red blood cells, and if it actually finds a problem, it can move out of the blood vessel and move out into the cell. And that was the inner life of a cell, full version. Okay, thank you for stopping by, and I appreciate it. Bye.